biology is technology. And what I'm gonna try and do is convince you guys that biology is no longer a science of discovery where we study nature and we toy with things and we tinker and we open them up and explore what's outside there. But we can actively engineer now and go through the design, build, and test cycles and make use of things which we need from our everyday lives and into the economy, broader economy. So before that, quickly, I have a very linear sort of journey. If you can see the logos up there, I've just pointed them out for you guys. I went to school in Riyadh. I was born to parents who were living in Riyadh, Saudi Arabia. So I was born and raised there. I went to school, finished grade 12, and then we decided to pack up and come to Pakistan. That's where I went to undergrads. We were in Peshawar. This is the University of Peshawar. It's second oldest in the country, pretty historical, huge campus, and the first ones to offer biotechnology those days in their undergrad. So that's why I went there, and this is the reason I didn't go to medical school, and I tried to convince my parents that I really want to do science and I want to be a scientist. Luckily, at the end of that, I got a place and I went to, and a scholarship, of course, a couple of scholarships. And I went to Oxford for 5 years. I did my master's, I did my PhD, DPhil as we call it, and then came straight back to Peshawar to do the things I really wanted to do. So these are the two things which I did when I came back. I built a social enterprise. It was a project, but it evolved into a full-blown startup, social enterprise. We have full-time employees now. And the startup's called Peshawar 2.0. And we work on engineering the local startup ecosystem in Peshawar. Peshawar's been really underprivileged and at the back of everyone's mind in policy and pretty much sidelined. A lot of youth, youngest province in the country, the youngest city, with a lot of youth, but still underprivileged and almost zero opportunities. So we came in and we said, let's engineer something. There's a lot of talent here, densely packed into the city, and whether we can manage to rewire and spark really cool things. So that's a different story, maybe for another time. But the things we're going to talk about today is the institute I founded 4 years ago. I chose a private sector engineering university, and I convinced them to go into biology. So now we have the Institute of Integrative Biosciences. I'll let you guys Google it and maybe check out the website, etc. But for me, what I'm really proud of, it enabled us to bring something as cool as synthetic biology into Pakistan. It's pretty new in the subcontinent. Not many people are working on it, but it's the trend currently that's happening in biology. That's the wave we're witnessing in biology. Whether you are a startup guy or whether you're an academic, I think it's pretty hot from San Francisco to anywhere in the world. So yeah, and it was not a research project, it was basically a government project. So I managed to sneak into the government and lobby and convince people, connect to people and convince them that this is really important for our economy. Let's gradually go back to high school biology and quickly come to speed in like 15 minutes from now on what's happening and what's at the center of it. That's more important. We really have to understand what's driving all the change and what's fundamentally changing, which wasn't there like 10 years ago, and it's there now. It's very cheap now in 2018, and that's what's driving innovation in biology and biotechnology. So this is my favorite diagram. If you can see it, I hope you can read it. It's an inverted triangle. At the top, you have things we call botany and zoology, basically whole organism biologies, where you look at animals and plants in their full sort of multicellular systems. And we look at like, think Nat Geo, think Animal Planet, and think all the sciences that go into that, observing how plants and animals behave, how they grow, how they reproduce, how they communicate, how they hibernate, etc., how they interact with the ecology. So that's whole organism biology. But as we got hold of a lot of technologies and science and biology basically evolved over the past couple of centuries, we got into smaller things. So if you look at the diagram to the side, we have things which are which we can see with the naked eye, but then we have the microscopes. So now we can go deeper into cells. And basically this is what enabled biologists, which were naturally reductionists. They like opening things and looking what's inside. So basically they opened organisms up and they found lots of organs. And that's how anatomy and physiology came about. And then they started opening up organs to see what they're made of, and they found tissues and cells. And that's how the cell was discovered in animals. So we got cell biology. Then we also in parallel got to know what disease was, infectious disease. And there were small things which were causing them, and then we named them bacteria, bacterial cells, etc. So basically technology and science evolved, and it enabled biologists to dig deeper into living systems. So upper you have animals and plants, and as you go deep, you have cells, and if you go deeper into cells, you get molecules. And the two most important molecules we have in biology, this is something you need to note, is our genes, and then we have proteins. Genes are made of DNA, so basically it's DNA and proteins. These are the most important building blocks in any living cell. DNA is the software in an engineer's language. Proteins are the hardware. Proteins do the job. DNA carries the information, the code for all those jobs or functions. And one last thing to note over here is the DNA, the language, the code, the software of life is 4 alphabets. And it's the same across the entire living kingdom. So the animal and plant kingdoms. So all living organisms are encoded for by the same DNA molecule, and most of them have similar proteins which are conserved across species. So basically, the entire diversity of species at the top of the triangle here, be it 1 million, 5 million, whichever number you pick, all those species kind of unite at the bottom over here in molecular biology because they're made up of the same code and the same hardware. And obviously, if you have the tools now all of a sudden everyone's interested in looking at the DNA and the protein of their subjects. For example, a crop guy would want to check out wheat. A doctor wants to see how his cancer patient is different from a normal guy. An animal guy would be interested in why my cows are producing less milk than the other species, for example. I want to check out the code. So now all of a sudden, everyone's interested in the molecular biology, the bottom of this triangle here. They're interested in the molecular biology of their living system. So to summarize, biology's evolved from whole organism biology, and now we can dig deeper into cells, and we can go beyond cells into different parts of the cell and different building blocks of the cell. That's what we call molecular biology. But if you look at this picture over here, this is a very, from an engineer's perspective, this is pretty startling because it's there's one piece of information which is really crazy out here. So basically, if you ask an engineer that this complex little machine over here self-assembles from this cell, it's literally unbelievable. So how can a machine or a robot self-assemble? But here we have a complex organism self-assembling from this first little cell over here. And then the next logical question is, where's the manual for this assembly? Where's the code? And if you narrow down on it, obviously over the last century, scientists found that it was the genetic material inside the cell, and within the cell, inside the nucleus, this pink thing you see in the middle. And once they started studying it, they found these guys inside the nucleus. And they said, this is the genetic material. This is what we call the chromosome. So basically, when cells divide, the long stretch of DNA threads are rolled into these really nice, tidy chromosomes so they can divide easily. But when you open these guys up, if you look at this diagram over here, starting from the bottom, if you uncoil a chromosome, you come across a lot of pink proteins here. And then if you still uncoil them further, you see the double helix of the DNA. So basically, this is the point where we arrive at the fact that this thread-like thing, this thread-like chemical, this DNA molecule, is responsible for storing all the information for this guy to self-assemble. And in a very layman's language, the code of life is right here encoded in this stretch of DNA. And then again, from an engineer's perspective, the code is a sequence of 4 different types of molecules. So the rungs of this ladder as you see them. And basically, if it's just a sequence of 4 different letters, if you abbreviate them into letters, A, T, G, and C, the entire DNA now, all of a sudden, can be saved in a text file. So all of a sudden now, biology from being whole organism over a period of a century evolved into molecular biology. And now all of a sudden, after 30 years, 3 decades maybe, it's become digital. So now we no longer need to go into the jungle and study the characteristics out there, but you can start to tap into the actual software of life, the text file of life, and see if you can get any signals from there. And the more we know about the genotype-phenotype relationship, the relationship between this code and the proteins out there, and the resulting functions out there, the more we can predict. So now all of a sudden, and this was like when I was a student, this was like 2001, 2003 was the first draft of the human genome when it was published. And it was like just like what, 15 years ago. So it was just 15 years ago when biology suddenly became digital. Now you have the code of life for almost any species that we work with or that we consume or that we use in the industry. Okay, so and this is just Matt Ridley's sort of description, a layman's description of what it looks like. If I read to you the whole genome at the rate of 1 word per second, 8 hours a day, it would take me a century. In very layman computer terms, the entire genome is about a text file which is about 40 gigabytes. So if you want to save a human genome on your external drive somewhere, it's going to take 40 gigabytes on the hard disk. More scientifically, this is the growth rate of DNA sequence data in one of the largest public databases. That's called the NCBI, run by the National Library of Medicine in the US. So this is all publicly available, freely available genome, DNA, and increasingly genome databases, data sets in this database. So as you can see, it's pretty steep. The growth is crazy. It's exponential, and literally we have more and more data sets and less tools for analysis and digging deeper, gaining clinical or biologically significant insights from this data. So the task is becoming heavier. Another anecdote people share is storage is an issue, let alone analysis. Like the storage is an issue. Data is not an issue. It's the storage and the analysis now being the bottlenecks. So life all of a sudden is a text file. Now we can start playing with it. So the first attempt was in this little cute organism. I don't know if you guys can guess what this is. Can I take one guess? Anyone? Are you guys with me?
Is it amoeba or this? Is it a cell, Faisal?
It's an insect. So Can you guess this?
Oh, that's, oh, I know what it is. Is it, which one is this, Anya? Is this, let's see. Oh, I know what. Is it that, what do you call that, that ladybug or something?
Firefly.
No?
So it's a jugnu.
Oh, Jugnu, correct. Yes, yes. Oh my God, yes.
So it's a
Gotcha.
firefly. Okay,
Uh-huh.
so what the scientists thought was, can we take the code for this this glow in the belly of the jugnu, the firefly, and maybe take the code and paste it somewhere else in some other organism and see if that can glow? So this was like a copy and paste experiment within the text file. So what they did is they took it and they put it in a plant, out of all things. And when the seed germinated, this is what they got. So this was like proof of concept that you can literally take the code for the glow, which is the luciferase gene system. You can paste it somewhere else, even in an animal, in a living organism, which is as far as a plant, like taking a code from an animal, putting it in a species of another kingdom, a plant. It still glows, it still works. So the language is universal. Animals, plants, microbes, all are made up of the same software. And this was proof of concept that you can take the code across kingdoms, let alone species. So this is 1970. This is genetic engineering. These were the first copy and paste experiments. Pretty expensive, very expensive, but again, we had results. One quick application, my favorite one is, so this is Artemisia, the plant, which is found in like Nepal or North India, North Pakistan. Chatral is the place in Pakistan. Uh, but this guy produces an anti-malarial drug, artemisinin. And the problem is that artemisinin is not found in Chatral or Nepal. It's found in Africa. So now how do we take these rare sort of herbs into Africa and make use of them? So what they did is they took the code and put it in the yeast, which is this thing in the middle. These are small yeast cells under a microscope. Yeast grows anywhere, everywhere, at room temperature, doesn't need any luxuries, any controls, and the cost is really low. So the result was they produced tons of artemisinin, and they now obviously save a lot of lives in Africa. So this was one really cool impact where you take a code, put it in a different organism, and put it to use at a significantly lower cost. Okay, so this was 1970, but what's so different now in synthetic biology, and what's taking us to the next generation of genetic engineering? I don't know. I don't know if you guys. I'm sure some of you guys know about Moore's law. This is Moore's law. Gordon Moore was the co-founder at Intel, and he predicted that transistors are going to double in performance, double in count in microprocessors every 18 months or so. And this sort of steep growth curve tells us how transistors and computers evolved. So in layman's terms, this is Pentium, the machine I bought when I was a kid, or my dad gifted it to me maybe in grade 5. But since then, obviously, you see all these things develop, and the end result is the computers have reached our pockets. So we have stronger, more uh, 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 high performance computers in our pockets than these guys over here. But what I want to compare this to, this really cool development in computer science, in chips and transistors, which has no doubt changed our lives for forever. I'm going to compare this graph to this one. So over here you have Moore's law, the blue worm over here, the blue line. But take note of the red line over here, which crosses the blue line and which goes steeper and higher than the blue line, and which basically represents productivity in DNA sequencing and the power to read DNA, which obviously underlies all the things we do in biology. So how easily, how cheaply, how accurately can we read DNA, the software of life? So this worm is now telling us that we're pretty much beating Moore's law. So over here, it's 13 times Moore's law. So biology is kind of, if you generalize, biology is developing 13 times more rapidly than computer science, which is pretty startling. And this is pretty much the dawn of a new age in biology, where you can so easily and so cheaply read the DNA, the entire DNA of any living system that you wanna look into. This yellow line, which is lagging, is another very cool technology. This is DNA synthesis, which in layman terms is writing DNA. So can we print off DNA? The red line tells us we can read it, but can we actually write it? So we can feed it to a bacterial cell so it can start listening to those commands or those or executing that software, whichever an analogy you want to use. So the red line has already outpaced Moore's law, and the yellow one's catching up. If we look at the costs, the blue over here is DNA sequencing, and as you can see, it's exponentially going down. And these 5 years were extremely steep, as you can see over here. So from a price tag of about $3 billion, the first human draft, the human genome draft, took us 15 years and $3 billion. The cost has come this down exponentially over the past few years. This is the machine they used for sequencing the human genome, which cost about $3 billion. This is the machine we use now, and it costs us $1,000. So from $3 billion in about 15 years, the cost has come down to $1,000. Now that's breathtaking, that's crazy. And that's what's fueling all the innovation here and allowing youngsters to get access to all of this and play with it. So the story doesn't stop here, of course. This is Oxford Nanopore's product, the Minion. They launched it in 2014 and they're selling it now. It's like selling like hotcakes. And basically it's a USB stick size DNA sequencing machine. So you can carry it around, costs you about $1,000. And you can read DNA in real time. So even if you're just a pet enthusiast buying a new horse or something, or a dog, and you want to check out the species, you can literally do that in real time. So different folks are trying it out in different scenarios and publishing their results. And Oxford Nanopore recently announced they're going to do a similar one pretty soon for your iPhone. So basically, you pull that little drawer over there, put a drop of DNA, slightly pre-processed DNA, and the phone reads it out for you, what's inside. So again, some breathtaking developments within the DNA sequencing technology, the technology which allows us to read DNA. Now similar stuff happening in synthesis. So for those of us familiar to this, we used to copy and paste DNA molecules from nature because we never really were able to print them, or we print them at scale. But now this synthetic cycle, this synthetic chemical cycle, it's a 4-step cycle. That's evolved so much now that we have these machines over here being developed, further developed at a very rapid pace, and miniaturized into things like these. So pretty soon we're going to have desktop versions of DNA printers. So now you have a USB stick of DNA sequencers. Pretty soon we'll have DNA printers. So imagine the level of innovation which this is going to spark and catalyze if youngsters, founders, startup founders in every corner of the world get access to all of this. So these little machines are already penetrating the fab labs and the maker spaces of the world. We've discussed this in Palo Alto very, very deeply when we were there last year in my cohort. But the DIY biology movement's already here now. So these rapidly declining costs are enabling all of this to happen. One last slide about costs. This is golden rice, where it was biotechnology, it was GM rice genetically modified rice, which had vitamin A as well, a precursor to vitamin A, and anyone who took it got extra vitamin A doses. That piece of research cost them $100 million back then in 2000. The same thing in 2018 now would cost you less than $5,000. So again, from 100 million to about 5,000 in 2018, it's pretty crazy. It's similar to that transistor moment where the homebrew club was playing with chips. They could produce small PCs, and that's why we see this is the next PC revolution that's going to hit us. Democratizing all this technology, taking it to the masses, enabling a lot of innovation, and taking all of us to the next level. So this is what we call traditional biotech and genetic engineering. And the sad thing is that in countries like ours, all these old professors are still evangelizing their own little piece of art, which was genetic engineering. But now things have evolved and we've got synthetic biology. And that's why I'm really proud that the government of KP in our province was able to be convinced, and they took this forward, and we're still building on it. So there are 3 trends. This is the third one. One was DNA sequencing, drop-in costs. Second was drop-in costs in DNA synthesis. And the third one is engineering principles, which is really close to my heart. So I always envied engineers all my life when I was a kid, especially. Why can't we build stuff like engineers? Why can't we design things, put those pieces together, and make them do things? And if you look at this analogy, the hierarchy is pretty strikingly similar in electrical engineering and in biology, in computer science and in biology. But the difference was that these guys could think bottom up, they could design bottom up, they could think of anything they want, they could do simulations, try it out on PCBs, and then fabricate those chips and go upwards. But the problem with us was that we could only go top down. We could only explore nature, just open it up, and have a sneak peek, and that was it. Which always kind of really annoyed me, that why can't we do more with this? But through synthetic biology now, through these 2 technologies that we have, through new approaches, which involves engineering principles, as I write over here, we are now able to flip the arrow. And for the first time in biology, we are able to design, build, and test things the way we want. So you think of any piece of DNA anywhere in nature, and think of mixing and matching all these properties, all these different functions, just like you would mix and match your electronic components on your circuit. Exactly in an exact similar way, we're trying to play with DNA parts, DNA components, and try and come up with devices which, if we put into cells, will function the way we want. So to summarize that analogy, we have design, test, and build in computer science, where we code and execute software and debug it and go back. In engineering, we draw these circuits and then fabricate these beautiful chips. In biology now, we design devices, we design genetic circuits, and then we print them, just like these little molecules. So this is a small little tube. The white fluff you see over here is DNA. It's a lot of DNA, but a lot of DNA to be seen with a naked eye. But again, that's printed DNA for you, and for you to take and put into cells, so the cells can start executing the code and do things you want. And we've stolen a lot of principles from engineering. So DNA looks like this. This is a normal bacterial genome, pretty complex, messed up, coiled up. What we want to do is turn this into Lego bricks. How can we break this into parts? Can we characterize them? Can we standardize them so they snap on to each other, just like Lego bricks, and they become interchangeable? And this is what's happening now. And we have an open source community now, again, borrowing from open source stuff in computer science, where we're building a huge repertoire of DNA parts, where folks are are sharing DNA parts under common standards, and they're able to mix and match and come up with circuits that they like. So again, abstraction, standardization, characterization, I'm not going to go into this, but basically we turn DNA into Lego bricks so anyone can start playing with it. So these are some of the circuits that people are building. If you can see, we're scribbling similar stuff as well. So basically in SynBio and synthetic biology, we aim to design and engineer bio-based parts or DNA parts, so we can come up with novel devices or circuits and systems, which are organisms. So you design something over here, you print the DNA over here, and then you put it into a bug so it can start following those lines of code. It basically thinks that it's its own DNA, and it starts expressing that and producing things according to that. But whereas it was actually ours, and now it's producing things that we want industrially or just for fun. This is a scaled up experiment. This is the first living cell whose parent was a computer. So this was a bacteria whose entire DNA was synthetically produced, designed using a computer, and then printed off, and then added back. And once it was added back, it rebooted into a different species. So again, the first piece of evidence for synthetic life, where you can produce, you can reboot an organism using synthetically produced DNA. We also now have logic gates for the electronics people out there. Gates made up of DNA circuits. So basically, more intelligent genetic circuits where it's taking inputs and giving you an output. We're also kind of coming together under a single language, so we can stop confusing each other with the language we're talking and the diagrams we're producing. So we also have an open language now. So basically, a cell used to be a factory, and the best you could do is maybe make it produce artemisinin or insulin or any other sort of drug. But now it's becoming a machine, an intelligent machine, because it has gates, and it has takes inputs, gives you outputs. It's autonomous, of course. It's living. And then it's self-assembling as well, which is pretty scary. You don't see self-assembling machines. Scary and exciting, of course. And then it's self-replicating as well. It reproduces. So once you hack a bacterium, a cell, it's going to reproduce if you provide the right environment. You don't need to make copies of it on a conveyor belt. You just grow it in a culture medium, come back tomorrow morning, and there are millions of them. So again, self-replicating, self-assembling, autonomous and intelligent. I'll end with these examples. So I don't know this guy, and I'm not an LA Galaxy fan, but I like his tie. So the tie he's wearing, the necktie, is produced by Bolt Threads. You guys should follow it. Bolt Threads are people who are making yeast cells produce spider silk. So the tie is made up of spider silk over here, thanks to synthetic biology. And they're managing to spin out fibers out of these yeast cells, thanks to the code from spiders. Adidas has adopted similar technology, and similar fibers, and they're doing this shoe called BioSteel. It's made up of spider silk. Similar material stuff is happening between Spyber and North Face, and they're doing these jackets with really cool properties. These are biosensors, which are cells that detect things. And if you think of sensors, and if you fast forward, you had sensors, and then all of a sudden you had Internet of Things. These are biosensors, and now we're thinking of the Internet of Living Things. So if these guys can take inputs and give us outputs, these cells, the pink ones over here, they detect carbon monoxide, and they give out a red color. So now, can we patch these guys up? Can we have an Internet of Living Things? These guys, Impossible Foods, have been doing meat. So this is vegetarian meat, which bleeds, which sizzles, which feels like really proper meat. And again, this was news, by the way. Yesterday, they got approval from the FDA. So you're probably going to see this in the market pretty soon. This is an image from the rover. I was going to ask you guys, but there's no time, I think. So they plan to send out one of those USB sticks to sequence the surface soil, if you call it, at Mars, to see if there's any living creatures. And if there is any living creature, any living cells, they should have DNA. And if there is DNA, it could sequence that real time and email us back any sequence. So these are experiments in the pipeline to look for Martian cells on its surface, which is pretty exciting, all based on DNA sequencing. How much time do we have, if I can ask?
Faisal, actually, we are almost running out of time. So if
okay, yeah, I'll take half a minute. So
sure.
I was just gonna explain or maybe describe what we've been doing in our lab. But these are two iGEM teams that we had in our lab. iGEM is the International Genetically Engineered Machine Competition, genetically engineered machines. And it's a SynBio, synthetic biology competition that happens in Boston. So we sent out teams in 2016, won us a bronze medal in the bronze category, which was pretty cool. Peshawari boys and girls from Pakistan winning us a bronze medal. And then last year we sent out a team which did a biosensor for arsenic in freshwater. I was going to talk about Indo-Pak and the Indus, but we don't have any time. They try and engineer a fish which would change color if there's arsenic in water. They also did a small bot, Arduino-based bot, which would text us if the bacteria detected any arsenic. So again, an Arduino bacterial interface earned them a standing ovation at the competition, which is pretty cool. I had a video as well, but you can check this online. This was the team. Quickly, these were some YC quotes and quotes from Bill Gates and Steve Jobs about biology, but I'll fast forward. So biology is molecular. Biology is now digital all of a sudden, and it's entrepreneurial. More and more people are getting access to it, and people are toying with the idea of engineering living systems for industrial applications and all sorts of other applications. I was also going to describe my pivot these days. So I've relinquished the directorship at the Institute. I've hired a full-time director at Peshawar 2.0, and I'm in the process of building Aviros now. Definitely Pakistan's first synthetic biology startup, perhaps maybe even an Indo-Pak startup, because I don't hear anything in India as well. So fingers crossed. I'll stop over here. Thank you all for listening.
Faisal, can you guys hear me?
Yes, Ashra.
Yeah.
So I have a question. If people don't mind, I think Faisal just mentioned something about the Indo-Pak water or something, and I'm wondering if we can let him speak about that for a couple of minutes. Is that okay, Shaivi?
Yes, Ashra.
Yep.
Okay, do you want to just tell us about that also, Faisal? I'm curious
so
what that is.
Yes, so the iGEM 2017 project was inspired by this graphic from a science paper, which basically shows the Inder and its neighboring rivers in branches in red. And basically, it has high arsenic content. And remember, all of these water channels or these tributaries of the Indus come from neighboring India. So God knows what the arsenic content is in neighboring India. I don't know if there are any studies. I'm ignorant of that. But again, arsenic is a pressing environmental issue.
Faisal, can I ask you a question about, apparently the new PM Imran Khan is talking about, starting some collaborations, right? And I'm wondering if it makes sense for you to bring this topic up and perhaps give a talk about this, right, in India?
I would love to. Yep.
Okay, so all of you, please apply for your visas, okay? Because I'm speaking on November 30 and December 1 at Thai Global in Delhi. So I'm actually keynoting. So if anybody is free, I think you should come. It'll be, it'll be really quite an eye opener. And Faisal, you should talk about this point. I think it will be amazing if we can independently start off a collaboration. I don't think we need to really get anybody's permission. You know what I mean? And
That's true.
people will be happy to see that we are doing it on our own.
That's true. Yep. I think that's the RC spirit.
Yep. Okay. All right. Anybody else, guys? Please feel free. Sorry, I'm hogging up.
For giving us such great insights about how biotechnology are, together. Please ask questions, everyone, if you have any. I know we are almost, over, but still we can have some quick Q&A quickly if you have any questions.
Hey,
Actually,
Ali here.
Hi,
yes,
Ali.
Ali, please go ahead.
Okay, yeah. So, Faisal, thank you for the presentation again. I think it was a great summary of what we wanted to know and could have known, I guess, in this time. So, a question that I've been sort of thinking about recently, especially after reading a few sort of doomsday scenario kind of articles, is about sort of the whole biohacking movement and a lot of people working with these sort of reduced price kits at home. And people have their sort of biology labs, DIY biology labs in their basements or garages. And just sort of thinking of how any new drugs or any sort of synthetic manipulation that's done in any organism in a traditional sort of lab has to go through 500 checks and maybe years and years of different kinds of tests to get anywhere. So there are obviously a lot of people sort of talking about the possible sort of worst case scenarios with biohacking. So what's your take on that? Because of course, I'm from knowing you, I know that you're excited about that movement as well. So how do you sort of maybe align those fears with the excitement of it?
So the quick, easy answer is obviously the fear is there because it's new. I think similar to any other dual-use technology, even computer science, like we never knew there were going to be viruses, computer viruses, and the losses, and then fast forward into fake news, for example. So for every smart bad guy, there will be tons of more smarter cool guys, right? So with every threat, there will be equally maybe more clever solutions for that. And I think only time decides. As the industry aligns, as standards emerge, as standard practices emerge, as safety regulations emerge, I think things will evolve towards the better. There hasn't been any incidents so far. There have been very limited cases. And even in the US, the policy is to allow and accelerate DIY biology and its movement, and not stifle or regulate it, because the benefits outweigh any risk.
Thank you.
No problem. I think there's a mic unmute which is pretty noisy. If there are any other questions, I'd love to take.
Hi, Faisal. This is Fariha Anwar from Impact Network. I had a question. I wanted to ask more about the applications of synthetic biology. I can only imagine agriculture and health at the moment. Do you think it has applications in other sectors as well?
Yeah, so the bioeconomy is pretty huge in Pakistan and India, especially. You have health, energy, livestock, fisheries, forests, chemicals, materials, food, feed, all sorts of stuff. but obviously agriculture is the biggest one, being an agriculture country, and health is another big one, being a very maybe unhealthy country, whatever you may call it. So yes, so those are the two big sectors, but again, all these sectors benefiting from living systems will benefit from synthetic biology.